Sterility testing is a critical safeguard across pharmaceutical, research, and clinical laboratories. It protects product integrity, ensures regulatory compliance, and ultimately safeguards public health. But when sterility testing produces false positives, the consequences can be significant. Delayed projects, increased costs, lost credibility, and unnecessary investigations. Understanding why false positives occur and how to prevent them is essential. Sterility testing goes beyond confirming the absence of microorganisms. It's a controlled, highly sensitive process designed to detect even trace contamination. Common methods include membrane filtration, direct inoculation, environmental swabbing, and biological indicators. In highly regulated environments, even minor procedural lapses during sample preparation, handling, or incubation can lead to misleading results. That's why mastering the fundamentals is the first step toward reducing false positives. False positives most often stem from contamination introduced during testing, not from the product or environment itself. Common causes include poor environmental controls, improper aseptic technique, inadequate personal protective equipment, contaminated tools or residual chemicals, and errors during sample transfer or handling. Trust in sterility testing starts with the tools themselves. Whether using swabs, contact plates, or biological indicators, every validation tool must be proven, not assumed, to be effective. For biological indicators, this means a known consistent spore population, highly resistant organisms such as Geobacillus stérothermophilus or Bacillus atrophius, documented resistance values or D values. Strict aseptic practices, hand hygiene, sterile equipment, controlled airflow, and cleanroom discipline are essential to prevent cross-contamination. Operating within laminar flow environments and maintaining validated cleanrooms dramatically reduces the risk of introducing contaminants during testing. Disinfection processes must be both effective and consistent. Facilities should verify that disinfectants are EPA registered, validated in real-world conditions, free from residues that interfere with testing. Using biological indicators to validate disinfection cycles ensures contamination is truly eliminated without generating misleading results. One of the most powerful ways to reduce false positives is ongoing staff training. Regular education reinforces aseptic technique, updates teams on new technologies, and builds confidence in identifying contamination risks early. Audits and competency assessments help identify gaps and reinforce a culture of precision and accountability. Biological indicators prove Curis Decon is reliable and repeatable at greater than 6-log efficacy every time. Sterility testing equipment like incubators, autoclaves, and filtration systems must be routinely maintained and calibrated. Even small performance deviations can compromise results. Preventive maintenance is not optional, it's a critical safeguard. Rapid microbial detection technologies are changing sterility testing. These systems reduce testing time from days to hours and provide clearer insight into contamination sources, helping eliminate false positives while improving efficiency. Tracking trends over time allows laboratories to spot recurring issues and correlate false positives with environmental or procedural factors. Robust data monitoring not only improves accuracy, it strengthens regulatory compliance and operational confidence. Robust data monitoring not only improves accuracy, it strengthens regulatory compliance and operational confidence. Sometimes the fastest path to improvement is external insight. Consulting sterility experts helps labs troubleshoot persistent issues, refine protocols, and stay aligned with evolving best practices. Reducing false positives in sterility testing requires precision, validated tools, disciplined technique, trained personnel, and smart technology. When these elements work together, laboratories gain confidence in their results and trust in their processes. Accuracy isn't accidental. It's engineered every step of the way.